Ido and Michael for organizing very stimulating sessions and SIG. Today I talk about the application of text mining on finding potential moonlighting proteins from PubMed text information. This is done by my former student Ishita-kan and my collaborator Mansoor Bui Yan and from IUPUI Indianapolis and me. So let me first introduce what moonlighting protein is. Moonlighting protein is a protein which does the two or more independent functions. And in strict definition by Jeffrey, those two functions are not occurring because of the multiple domains or splicing variants or gene fusions or the situation that the protein does the same function in a different pathways. And more, it is likely that the protein had an ancestral function, original function, and then gained the function, additional function along the way of the evolution. Now, most commonly observed moonlighting protein is that they are enzymes, and which are well-known enzymes, and happen to found in a recent large-scale screening that it binds to DNA or inhibits some expression and so on. This is an example how the moonlighting proteins or moonlighting function can occur. This is taken from Constantine Jeffrey's review article. The A is showing that moonlighting function can occur by different localization of the protein. B is showing that example that protein inside and outside of the cell. Okay, again, this is a localization of the protein is affecting their functions. Or BC's expression in different cells, or the other things includes that binding different cofactors or oligomerization states and so on. And here is some examples of the moonlighting proteins. The first one is aconitase. This is an enzyme in TCA cycle, but it is also known as a function 2. It is also found to be involved in iron homeostasis. And the function will change by the change of the concentration of the ions. And well, as you see, some other examples. Well, let's see. Like the third one from the bottom, glutamate racemase. This is well. This is also known to be involved in DNA gyrase inhibitor. So actually, those moonlighting proteins are found by chance, by additional later experiments, but now accumulating in the databases. The first one is MoonProtDB. This is collected by Jeffrey in Chicago. This is collected by manual curation of PubMed databases by reading the literature. Second one, multitasking protein DB. This is from this is collected from PubMed by keyword search. Well, keywords such as multitasking or moonlighting or bifunctional, and those kind of words are used to mine the search PubMed databases. The last one, MoonDB. MoonDB is done by Brun Lab, and what they did is they collected first. This is concentrated on human moonlighting proteins, and this is first they did a literature search for finding a human moonlighting proteins. And in the paper, they also did a network-based prediction of moonlighting proteins. Idea of this network-based prediction is, starting from protein-protein interaction data. And if you see a protein is interacting with, very naturally, different type of functions, protein with different type of functions, locating at the intersection of two type of functions, then they tend to be a moonlighting proteins. But so there was those effort collecting databases, but the number of moonlighting proteins, especially well curated, I would say the MoonProtDB on the left by Jeffrey is probably most well curated moonlighting protein database. The number of these proteins moonlighting proteins are about a couple of hundreds, and it does not increase that much because it needs a very careful curation of the literature. Now, okay, but it looks like there are more and more moonlighting proteins in various genomes. So we have been working on trying to develop methods to find potential more moonlighting proteins, because if we have lots more moonlighting proteins, we can do lots of bioinformatics stuff. Bioinformatics needs data to do many things. Well, first natural idea is to go to the UniProt database or PubMed database and then see the current annotations. Well, in UniProt, there are moonlighting proteins or bifunctional proteins, but they are not all of them are labeled as moonlighting or dual function or multitasking. So if you just do the keyword search, well, you can get some, but there are lots more hidden potential moonlighting proteins in the database. Now, then first approach we can think of is, well, because UniProt has a Go-term annotation, can I see the Go-term annotation to classify the Go-term function and then find the dual function or multiple functions? Second is, well, the UniProt has literature information, text information, well, the title of the literature, abstract of the literature, and the functional description curated by UniProt team. Can we develop text mining methods to find moonlighting proteins? Another approach you can think of is well, in case that protein is not well curated, does not have much experimental information, can we go to the omics data, like protein-protein interaction data, expression data, or some other data, and then predict if it is a moonlighting protein or not? So actually, in the past, we have worked on the number one, the Go-term annotation, Go-term classification-based approach, and then also the last one, the large omics data-based approach. And then this time I talk about the text mining-based approach we recently did. So first I just in this just a quick slide, I show how we did the Go-term-based classification approach. It was done in 2014. It's already like 3 years ago. So what we did is we went to the UniProt database and collected Go-terms of E. coli, all E. coli, and then because it was moonlighting protein has not much studied by bioinformatics approach at that time, we are wondering if just simply clustering the Go-term functions by FANSIM score can identify moonlighting proteins or not. What is shown here is, so for a set of Go-term for a protein, I classify the Go-term based on the FANSIM score, and then x-axis is changing the cutoff value of the clustering, and then y-axis is the number of clusters. Of course, as I use a more strict cutoff, then I have more clusters. But if the protein have, even if the protein have lots of Go-terms, if it's only targeting only one type of function, then the number of clusters does not increase. So that's a hypothesis that this approach could potentially identify the moonlighting proteins. So this is the criteria we tried for the moonlighting proteins. Now, we also need to find non-moonlighting proteins. This non-moonlighting protein is always tricky. It's similar, like if you have worked on predicting protein-protein interaction, for example, it's very difficult to get the negative data, but at least we can, this is a temporal the criteria we used to define non-moonlighting proteins. So essence is that it has lots of Go-terms, but the number of clusters does not increase, even if I use a strict cutoff. Then by this criteria from E. coli, we found 140 potential moonlighting proteins and 150 potential non-moonlighting proteins. But then we read those literature, 290 literature or a little bit more because some protein have multiple literature, and then confirmed that at least 43 of them seems to be the new moonlighting proteins. But out of them, well, if we apply one domain protein have multiple function, this domain number, then it will reduce to 33. This is first thing we did in 2014. Now, last year, we also worked on omics database-based approach. So here, we used Go annotation and protein-protein interaction network, gene expression profile, and so on to predict the moonlighting proteins. Basic idea is that well, if a protein is interacting from let's say, as an example of protein-protein interaction network, if a protein is in the protein-protein interaction network, if it is interacting with different type of function, they are more probable to be moonlighting proteins. Same thing for like a gene expression pattern. If gene has same expression pattern of multiple different functions, then they may be a moonlighting proteins. So we fed it to a machine learning method, SVM and so on, random forest and so on. And then this is the accuracy with different combination of the omics data. So if you have a Go term annotation, it's very, very high accuracy. But if you have the other features, it is about 60% to 70%. Now, at that time, we also applied this to genome scale to estimate the number of potential moonlighting proteins. We applied to yeast, C. elegans, and human. Well, some of the proteins do not have the omics data, so we could not apply this approach. But okay, so at the end, we estimated about 10% or well 5 to 10% of the genomes in these three organisms may be moonlighting proteins. We didn't do we listed all those our predictions, but well, we don't have a experimental group in my lab, so we haven't confirmed it. But this is our estimation at that time. Now, this year, we try to do the text mining because it's very natural approach to go to the PubMed database and then find the potential moonlighting proteins. Data set we used is 263. This is taken from MoonProt database. Non-moonlighting proteins, this is the criteria, the non-moonlighting protein criteria applied to human, E. coli, yeast, and mouse. And then text information are taken from UniProt. And there are three types of text information I got. One is just title of the papers. And also, second one is abstract from those papers. And then third one is a function description in UniProt database. This is a number of titles, and in parentheses, this is showing the number of proteins which have those features, because not all of the moonlighting proteins had abstract information, and sometimes, well, the abstract could not be automatically retrieved by our web crawler. So that's the reason why this the number is decreasing from the number of titles. This is a number of proteins which have good size of the text associated to the entry. This is showing the distribution of the abstracts available in moonlighting proteins and non-moonlighting proteins. Most of them do not have so many papers, but and some of them have very large number of papers, 30 or 50 papers. Non-moonlighting proteins sometimes have more papers sometimes. Well, this is probably because that to identify non-moonlighting proteins, we wanted to really make sure that it is annotated with more than 8 Go terms, which means that there should be some literature behind it. So that may be reflected. That was probably reason that I have some peak here. All right, so this is a workflow of identifying potential moonlighting proteins. It consists of two steps. First step is text level prediction. Okay. All right. So each protein have different set of titles, abstract, and functions. And then for each of this text, I make the feature vectors by language models, the neural network-based model, the back-of-word model, or topic model, and then feed it to the classifiers, logistic regression, and SVM and so on, and then make the prediction at the text level first. In the second step, because some protein have multiple texts, then I do the voting. If a protein have, let's say, 5 text information, I do the function, well, moonlighting protein prediction for each of the 5, and then do the voting at the end. 3 language model I used is the TF-IDF. Hagit has gave a very nice the introduction of this approach. This is counting the number of words and estimating the importance of the words. And then there's another method called latent decay allocation. This decay allocation is representing a text by probability of the topics. The last one is the paragraph to vec. So this gives the unsupervised vector representation of each text information. So all these methods will give the vector representation of the abstracts or the titles, and then I fed it to the machine learning frameworks. Those are the hyperparameters. I don't go to the detail, but those are the searched by grid search. Then I did the 5-fold cross-validation. 3 subsets are for training. 1 subset is to identify the best hyperparameters, and then 1 subset was for testing. And then the value I'm reporting is average over the 5 test data set. Now, okay, so this is the text level accuracy. So this is okay. So this is the results of using titles, abstract, and function description. And then these are the 4 language model. Okay, there's two neural network-based feature representation. Deep is taken from the text information, taken only from moonlighting proteins and non-moonlighting proteins text. PDeep, this is pre-computed deep learning feature vectors. This is taken from whole uniprot text information. And after those language model made features, those are the classifiers I applied. Linear regression, random forest, SVM, and GBM. So 93% was F. This not the percent. This score F score of 93%, 0.937 is the best the value I got. So actually, well, this is the second best, all done by SVM. Well, interestingly, for text and abstract, because probably the text is very short, just using TF-IDF seems to be working well. We don't need the network-based feature representation. Now, another thing to see is if I compare deep and the language, the feature vector from MP and non-MP against a whole uniprot text information. Actually, if I use whole uniprot text information, the feature vector looks like it's losing some specificity and didn't make an improvement despite of the loss of computation. Now, this is a protein level accuracy. Well, again, so this is the best value I got. So F score of 0.918. This is the second best. And then these are again, for titles and abstract, the simple TF-IDF method is working fine. And now, okay, so then, well, then at last, we'll again run these methods to whole genomes. And to do that, I use these 4 top methods, and then run each of them to genomes, and then took the consensus. And this is well, final results. These are, okay, we apply to East human, and this is the frog. Well, we use this frog because this is less annotated and do not have much omics information. Those are the coverages showing how many proteins can be analyzed by text mining methods, because some protein obviously does not have any text associated with the entry. So if I use, okay, I have 4 methods, and by voting, if I take the 3 proteins or more, 3 votes or more, then I have about 20% to 30% for Eastern human as moonlighting proteins. If I take vote of 4, all consensus, then I have about 10%. And well, if I compare with the previous approach we developed, which uses omics-based approach, it's similar. So about 10% is a current assessment of the moonlighting proteins in genomes. In summary, so we developed a text mining approach for identifying potential moonlighting proteins. And then, well, because the accuracy seems to be high, this can be used as a first filter before you manually read the papers to really identify the moonlighting proteins. This is an estimation, but nevertheless, this is really limited to the literature information which are currently available. So we need to take into this account. Okay, so this is done by Ishita-kan, who graduated last year, who was a very who was working on CAFA2 in the but now she's at eBay. And this is actually her husband at IUPUI, and he's now at IBM. Thank you very much.
Thanks very much, Daisuke. Any questions? Please come to the microphone.
There should be another one. Hi.
Hi.
Thank you so much for the talk. Super interesting. I had a question. Because there is a very low number of these moonlighting proteins that you have. And you showed this plot where you had the numbers of articles addressing moonlighting and non-moonlighting proteins. So for almost all of that plot, there were more articles on non-moonlighting proteins than moonlighting proteins.
Yes.
And that makes me a little wary, right? So it makes me wonder how high the resolution is of identifying the different functionalities, right? So is it really true that, you know, they studied this protein and these are really the 2 functions that they have, or 3 functional, however many functions they have, right? Or is it really just because maybe they're confused, or, you know, in some cases? And that, by the way, also sort of translates for the unannotated proteins.
Right. Yeah. So, well, yeah. So because the data is not large, well, there was always, yeah, I cannot, yeah. Well, there's a danger of the accuracy of this approach, because it certainly affect to the probably accuracy. Well, yeah. So I'm. Okay, yeah. I'm not sure how to solve this because the data is like this. So but in the current data set, I'm showing that it's probably possible to use this as a filter so that you can read it later by manually. Right. So I think we can because the framework is here. Maybe we can run this program every year or something, just like a CAFA approach.
I guess I'm just interested in why is it? You would expect if this thing has 2 functions, you would expect more papers, not less, right?
Oh, I see.
Can you speak in the microphone, sorry?
We need a mic over there. I'm telling you.
So it was my ex-team who did re-annotated the Go terms for the moonlighters from MoonProt. And what we found was there was a real psychological block. People were very clear, this is the function. And when a new function popped up, you could see people not accepting it.
Okay.
There is a real mindset in this. And also what we found in MoonProt is that there were things that were considered common knowledge, but actually when we went back painstakingly through the literature, there was no experimental evidence for it. It was just a belief.
Wow.
So be careful of the literature.
It's a little scary. Thank you.
Yeah, thank you.
So I was intrigued by the domain question and whether you've gone back and looked at these predictions for the multi-domain and whether it was a different domain
Okay, no, we didn't. For this work for using text mining, we didn't went back to the number of domains. In the earlier paper, we went back to actually PFAM database and then count the number of domains and separated the moonlighting proteins with one domain and multiple domains. But I don't remember if there was particular PFAM domains occurring frequently in the moonlighting proteins I found.
But it was more whether it's something to do with because the human proteins obviously tend to be quite large domains, and whether that gives them the opportunity to do more.
Okay, I haven't carefully looked into the number of domains.
Okay, well, I think we've run out of time, I'm afraid, sorry. Well, thank Daisuke again, and back here at 10 o'clock to hear the initial CAFA results.